Hello everyone, welcome back to my YouTube channel. In this session, we will be discussing about the process of glycolysis. So we know that glycolysis occurs in the cytoplasm of the cell. This is the process by which glucose molecules are split into two pieces called the pyruvate. So this first pathway is actually anaerobic, meaning it does not require oxygen. So it is the most evolutionary ancient metabolic pathways occurs in even the simplest of the cells. Right? Now here it is the steps of glycolysis. So first step, here it is the first step, glucose to glucose 6-phosphate. It is the phosphorylation step. First reaction is also called the hexokinase reaction. The enzyme hexokinase phosphorylates the oxygen on carbon 6 of the glucose to make glucose 6-phosphate. Okay, so the polar phosphate group traps the molecules inside the cell and also reduce the concentration of regularly glucose inside the cell, which encourages more glucose to enter by diffusion. This step causes one ATP, here it is the, which is provide the necessary phosphate group for the reaction. This is the second step, glucose 6-phosphate to fructose 6-phosphate. This is the step 2, it's called the isomerization. So glucose 6-phosphate isomerized to become fructose 6-phosphate. Okay, process which is catalyzed by enzyme, which is the phosphoglucose isomerase. Now third step, okay, which is called the second phosphorylation. It is. So this is the another phosphorylation, this time on the carbon 1 hydroxyl, which give us fructose 1,6-bisphosphate. This step is catalyzed by phosphofructose kinase enzyme, okay, kinase 1, and it will cause the another ATP. Here it is the. Now here is the first step, cleavage step. Okay. So now fructose 1,6-bisphosphate molecules is ready to be cleaved into two smaller ones, that is the GADP and DHAP. Right? Now fructose bisphosphate, here it is the adolase, this enzyme. Okay, adolase is lyase enzyme that will split fructose 1,6-bisphosphate into. Okay, so this is the adolase. They split the fructose 1,6-phosphate into the GDPA, GADP, sorry, or DHA. Here it is the. You can see this way. Okay. Right. Glyceraldehyde 3-phosphate or GADP and a molecule of dihydroxyacetone phosphate or DHA. Okay. Now here it is the fifth step. You can see it is the fifth step. Right? So fifth step, it is the conversion of DHA into GADP. The DHA will be converted into another molecules of GADP by the enzyme, which is the triose phosphate isomerase. Okay. So this is the end of the 5 step. Here is a 1, 2, 3, 4, 5 step of preparatory phase with 2 ATPs. Here it is this spent to achieve the 2 phosphorylation. Okay, 1 and 2 phosphorylation step. Now 6th step. Okay, here is the it is the oxidation, okay, phase. Now it's time for the payoff phase. Here is the payoff phase. One of our 2 GADP molecules from the preparatory phase, and we see the first thing that will happen is in oxidation. Okay, oxidation GADP to become the 1,3-bisphosphoglycerate. Right? Okay. This requires the NAD plus and free phosphate. Here is the free phosphate or inorganic phosphate, okay, to occur, and the enzyme involved is called the glyceraldehyde phosphate dehydrogenase. Here is glyceraldehyde dehydrogenase, okay, glyceraldehyde phosphate dehydrogenase. Okay, now 7th step, it is the It is the dephosphorylation step. Okay. So next, phosphoglycerate kinase, here is the enzyme, will catalyze transfer of phosphate group to ADP to become 3-phosphoglycerate, producing 1 ATP. Here is in the process. Since each of the 2 GADP, okay, here is all are the 2 GADP. Molecules will make 1 ATP, that is a total of 2 ATPs, here total 2 ATP, and for half the total payoff of glycolysis. 8th step, it is phosphate transfer step. You can say 3-phosphoglycerate. Okay, here is a mutase enzyme. Okay, phosphoglycerate mutase enzyme transfer the remaining phosphate from this hydroxyl to the next one over the make 2-phosphoglycerate. Okay. It is 9th step dehydration step. So the enolase enzyme catalyzes dehydration, resulting in the loss of hydroxyl group OH, okay, which will produce the phosphoenol pyruvate. Here is the. This is the last step or 10th step. Okay. Second dephosphorylation. Here is the. So the remaining phosphate group is transferred to an ADP. Here is the ADP by pyruvate kinase and generating another ATP and the pyruvate. So altogether it is a 10 step process. The first 5 steps, here it is the 1 to 5. Okay. Comprise the preparatory phase, which take 1 molecule of glucose and produce 2 molecules of GADP. Right. The will cost 2 ATP. The another phase, another 5 steps. Okay, here to here. Okay. Payoff phase, in which each molecules of GADP, okay, will be converted into pyruvate, producing 2 ATP. Okay, which 2 ATP each in the process for total 4 ATPs. Now remaining the net energy production from the 1 molecule of glucose is 2 ATP. So net energy production from the glycolysis, this is the 2 ATP. Certainly, the main thing to remember is that in glycolysis, glucose in the cytoplasm, here is a glucose, for the cell is converted into pyruvate. Here is the cytoplasm process. Okay, glucose converted into pyruvate. Now, the pyruvate, which will then move on to the next stage of cell respiration, which is the TCA cycle. Okay, so in next lecture we will be discussing the TCA cycle. Okay, this is all about today videos. Okay, I hope this will help you. Okay, thank you for watching.